Hello everyone, I am Mohammed Hamama, and welcome to your ASCP preparation camp. In this camp, we will cover all the topics on the ASCP lecture list to help you prepare for your upcoming exam. Today, we embark on the second leg of our exciting exploration into the fascinating world of the building blocks of life, amino acids, peptides, and proteins. But today, we'll be focusing all our energy on proteins, the complex workhorses that keep your body running smoothly. We'll explore their properties, how they're made, how they're modified, and their breakdown. We'll also peek at the proteins cruising through your bloodstream. Let's dive in. Proteins, the functional workhorses. Proteins are macromolecules essential for virtually every cellular function. From structural support in tissues like muscle and bone to the facilitation of enzymatic reactions and immune response, proteins play a critical role in maintaining human health. Understanding protein structure, a hierarchical approach. The functionality of a protein is intimately linked to its structure. Proteins exhibit a hierarchical organization, with 4 key levels. Primary structure, this level represents the foundation, consisting of a linear sequence of amino acids linked by peptide bonds. The specific order and type of amino acids determine the protein's unique properties. Secondary structure, here, the polypeptide chain folds into localized patterns, such as alpha helices and beta sheets, stabilized by hydrogen bonds. These patterns form the building blocks for the protein's overall 3D structure. Tertiary structure, this level represents the protein's three-dimensional conformation, achieved through further folding driven by interactions between amino acid side chains. These interactions include hydrogen bonding, ionic interactions, and disulfide bridges. The intricate folding process minimizes the protein's energy and optimizes its function within the cellular environment. Disruption of these interactions by factors such as extreme temperatures or harsh chemicals can cause the protein to unfold, denature, leading to a loss of function. Quaternary structure, for some proteins, additional complexity arises when multiple polypeptide chains, subunits, assemble into a larger, functional structure. Hemoglobin, the oxygen-transporting protein in red blood cells, is a classic example, consisting of 4 subunits. The arrangement of these subunits contributes to the overall function of the protein complex. Partners in function, ligands and prosthetic groups. Proteins often interact with other molecules to perform their specific functions. Ligands, which can be small molecules, ions, or even other proteins, bind to specific sites on the protein, influencing its activity. Additionally, some proteins rely on prosthetic groups, tightly bound cofactors essential for their catalytic activity. Hemoglobin, for instance, requires a heme prosthetic group to bind oxygen. Proteins lacking their bound partners are sometimes referred to as apoproteins. Now, let's delve deeper into the diverse physical properties of proteins that scientists exploit for various purposes. Light as a tool, protein spectroscopy. Certain amino acids within a protein molecule, like tyrosine and tryptophan, have the remarkable ability to absorb light at specific wavelengths. The amount of light absorbed depends on the protein's amino acid composition. This property, termed the extinction coefficient, allows researchers to quantify purified proteins using a technique called a 280. Additionally, some prosthetic groups, like the heme group in hemoglobin, also exhibit light absorption characteristics, enabling the detection of specific proteins. The power of charge, protein electrochemistry. Proteins possess ionizable groups that can be positively or negatively charged depending on the surrounding environment's acidity, pH. This electrical property significantly influences a protein's behavior and plays a crucial role in separation techniques like electrophoresis. By applying an electrical current, researchers can separate proteins based on their net charge, allowing for their purification and characterization. Solubility, a balancing act. A protein's solubility, its ability to dissolve in a given solution, is influenced by various factors, including pH, salt concentration, temperature, and the solvent itself. By manipulating these factors, scientists can selectively precipitate, cause proteins to come out of solution, or keep them in solution for further analysis. Historical methods like salting in and salting out utilize changes in salt concentration to achieve this. Additionally, fractional precipitation with chemicals like ethanol or polyethylene glycol allows the isolation of specific protein groups from complex mixtures found in biological samples like blood plasma. Size matters, unveiling protein architecture. The size of a protein molecule plays a vital role in separation techniques like size exclusion chromatography, gel filtration. This technique separates molecules based on their size as they pass through a gel matrix. Larger molecules elute, flow out, first, while smaller molecules are trapped within the gel for a longer duration. Ultracentrifugation, when spun at extremely high speeds, this technique separates molecules based on their size and mass. Denser and larger molecules settle out, more rapidly than smaller, less dense ones. Unveiling the mass, modern mass spectrometry. Modern mass spectrometry, MS, techniques offer scientists a powerful tool to determine the exact mass of proteins and their constituent peptides, smaller protein fragments, with high accuracy. This information provides valuable insights into protein structure and function. Beyond size and charge, advanced separation techniques. While size and charge are fundamental properties for protein separation, more sophisticated techniques exist. Ion exchange chromatography and isoelectric focusing, these techniques utilize the electrical charge of proteins to separate them. Proteins with different net charges bind to specific charged surfaces or migrate at varying rates in an electric field, allowing for their purification. Surface adsorption, proteins exhibit a unique affinity for certain surfaces. This property underpins techniques like reverse phase chromatography, where proteins with hydrophobic, water-fearing, regions interact with a hydrophobic stationary phase, surface. Affinity chromatography, this highly specific technique employs customized molecules like antibodies or other recognition molecules to capture and isolate desired proteins from a complex mixture. These recognition molecules bind only to the protein of interest, allowing for its targeted purification. Unveiling the chemical transformations, protein modifications. Beyond the basics, unveiling the world of post-translational modifications. Protein synthesis is just the beginning of the journey. Once proteins are built, they often undergo a series of chemical modifications that dramatically influence their function, location, and stability within the cell. These modifications, collectively known as post-translational modifications, PTMs, act like fine-tuning adjustments, ensuring proteins are optimally equipped for their specific roles. Fatty acylation, anchoring and signaling. Myristylation, a fatty acid called myristate can be attached to some proteins during synthesis, acting as a permanent anchor, tethering the protein to a membrane. Palmitoylation, another fatty acid, palmitate, can be attached to certain proteins after they're built. This attachment is reversible, allowing the protein to move within the membrane or interact with specific regions called lipid rafts. These lipid rafts are like specialized microdomains within the cell membrane, enriched in cholesterol and other lipids. Palmitoylation helps proteins interact with signaling molecules and other important players in the cell. An example is the hormone ghrelin, which stimulates growth hormone release. Only the form of ghrelin that has an octanoyl fatty acid attached can activate this process. This modification acts like a switch, turning on ghrelin's ability to trigger growth hormone secretion. Phosphorylation, flipping the protein switch. Phosphorylation involves attaching a phosphate group to specific amino acids within a protein, most commonly serine, threonine, or tyrosine. Our cells have dedicated teams for this process. Kinases, around 1,000 enzymes, add phosphate groups, while phosphatases, around 500 enzymes, remove them. Phosphorylation can directly affect a protein's activity, like flipping a switch. For example, phosphorylating glycogen phosphorylase regulates its ability to break down glycogen for energy. In some cases, phosphorylation can also influence a protein's location within the cell, subcellular localization. Phosphorylation of tyrosine plays a critical role in various signaling pathways, allowing cells to communicate and respond to external stimuli. Tyrosine phosphorylation often creates docking sites for other proteins, allowing signals to be relayed within the cell. The SRC family of kinases is a prime example, involved in transmitting signals from outside the cell to the interior. Interestingly, mitochondria, the cell's power plants, have their own unique kinase family that regulates metabolic processes. Here, phosphorylation modifies a histidine amino acid, influencing the activity of protein complexes involved in energy production. Prenylation, a helping hand for membranes and interactions. Proteins sometimes require extra help anchoring to membranes or interacting with other molecules. Here's where isoprenylation, the process of attaching a specific group, comes into play. Farnesyl pyrophosphate and geranylgeranyl pyrophosphate are the key players. These hydrophobic, water-fearing, groups are derived from cholesterol synthesis pathways. Proteins targeted for isoprenylation have a specific sequence motif called CAAX near their C-terminus, end. This sequence helps determine the specific isoprenyl group attached. More than 300 human proteins undergo isoprenylation, significantly impacting their function. This modification helps proteins associate with membranes, crucial for their role in various cellular processes, including signal transduction, as HRAS and KRAS proteins involved in cell signaling. Vesicle trafficking, as RAB2 and RAB3A proteins essential for transporting cargo within the cell. Cytoskeletal function, as RHOA and RHOB proteins that influence cell shape and movement. Nuclear membrane integrity, as Lamin A protein that helps maintain the structure of the nuclear envelope. Glycosyl phosphatidylinositol, GPI, anchoring proteins on the cell surface. Some proteins utilize a special anchor called glycosyl phosphatidylinositol, GPI, to attach to the cell membrane. The GPI anchor is a pre-assembled molecule synthesized in the endoplasmic reticulum, IR. Proteins destined for GPI anchoring have a specific signal sequence at their C-terminus, end, that acts as a docking site for the GPI anchor. The GPI anchor is transferred to the protein, and then the signal sequence is cleaved off. This leaves the protein firmly anchored to the membrane via the GPI anchor. GPI-anchored proteins may move more freely within the membrane compared to those with transmembrane anchors. They also tend to reside in specialized membrane regions enriched in cholesterol and specific lipids, called lipid rafts and caveolae. These regions might be important for certain cellular processes. Several essential proteins, like decay accelerating factor, DAF, and alkaline phosphatase, utilize GPI anchoring. Defects in a gene, PIGA, crucial for GPI anchor synthesis can lead to paroxys. Gamma carboxylation, a calcium connection for blood clotting. Proteins like factors 7, 9, and X involved in blood clotting rely on a modification called gamma carboxylation. Vitamin K acts as a helper molecule, cofactor, for an enzyme that adds an extra carboxyl group to a specific carbon atom, gamma, of glutamic acid within these proteins. A cluster of these modified glutamic acids is called a GLA domain. This GLA domain has a special ability to bind calcium ions. The calcium acts like a bridge between the GLA domain and the cell membrane, allowing these proteins to interact with and anchor themselves to the membrane in a calcium-dependent manner. Warfarin, a blood thinner medication, works by interfering with vitamin K's ability to activate these clotting factors. By preventing the formation of functional GLA domains, warfarin essentially throws a wrench into the blood clotting machinery. Glycosylation, adding sugars for function and stability. Proteins destined for secretion or those residing on the cell surface often undergo glycosylation, the attachment of sugars. These sugar attachments play multiple roles. Proper folding, sugars can help proteins fold correctly into their functional shapes. Export efficiency. Sugar modifications can act like tags that facilitate a protein's efficient packaging and export from the cell. Stability. Sugars can contribute to a protein's stability and protect it from degradation. There are two main types of glycosylation, occurring in different cellular compartments. O-linked glycosylation. Simpler sugar chains are linked to serine or threonine amino acids within the protein, typically in the endoplasmic reticulum (IR). N-linked glycosylation. More complex sugar chains are linked to the asparagine amino acid. A pre-built sugar structure is first assembled on a carrier molecule in the ER and then transferred whole to the protein. Once attached, the sugar chain can be further modified by enzymes in the Golgi apparatus, adding even more complexity. Hundreds of human genes are dedicated to glycosylation, highlighting its importance. Mutations in these genes can lead to various diseases. Sulfation, a sticky situation for protein interactions. Protein sulfation, particularly on tyrosine amino acids, is another PTM. Nearly 50 proteins, including blood clotting factors and hormones, undergo this modification in the Golgi apparatus. Sulfate groups are attached by enzymes using a specific donor molecule. The exact protein sequence that signals a tyrosine residue for sulfation is still being explored, but surrounding acidic amino acids seem to play a role. The exact function of sulfation is still being investigated, but it's believed to enhance protein-protein interactions, potentially influencing various cellular processes. For example, factor VIII, a protein essential for blood clotting, requires sulfation to function properly. Hydroxylation, adding flexibility and fine-tuning responses. Hydroxylation, the addition of a hydroxyl group, OH, is the most common PTM in human proteins. Collagen, a major component of connective tissues, relies heavily on hydroxylation of proline amino acids. This hydroxylation stiffens the proline rings, stabilizing the unique triple helix structure of collagen and giving it its strength and flexibility. Cells also use hydroxylation as part of a built-in response system to adjust to varying oxygen levels. A key player in this system is a protein complex called hypoxia-inducible factor, HIF. Under low oxygen, hypoxia, conditions, HIF helps the cell activate genes involved in processes like creating new blood vessels and red blood cells. However, when oxygen levels are normal, specific proline residues within HIF are hydroxylated. This hydroxylation acts like a tag, attracting an enzyme complex that targets HIF for degradation. By breaking down HIF, the cell essentially turns down the genes HIF was trying to activate under low oxygen conditions. Nitrosylation, a nitric oxide twist. Nitric oxide, NO, a small molecule produced by our cells, can also modify proteins through a process called nitrosylation. Nitric oxide attaches a special chemical group, nitrosothiol, to specific cysteine amino acids in thousands of proteins. The exact cysteine residues targeted and the effects of nitrosylation, activation or inhibition, vary depending on the protein. This process is still being explored, but it seems to play a role in regulating various cellular functions. Protein catabolism, breaking down to build up. Protein catabolism, maintaining a cellular balance. The amount of protein within a cell is tightly regulated. Just like keeping your home tidy, cells need a way to get rid of unwanted protein. This process, called protein catabolism, is the opposite of protein synthesis and ensures that only the necessary proteins are present and functional. Unlike throwing away trash, protein degradation is a very specific process that targets proteins that are no longer needed, damaged, folded incorrectly. This ensures healthy cells and efficient use of cellular resources. Protease power, the molecular scissors. The human body has a dedicated team of over 500 enzymes called proteases that act like molecular scissors, specifically cutting protein molecules into their building blocks, amino acids. There are 4 main types of proteases, each with a preferred way of chopping protein chains. Serine proteases, this is the largest and most versatile group, playing a role in digestion, immune response, and blood clotting. Cysteine proteases, these specialists are particularly important in programmed cell death, apoptosis. Aspartyl proteases, these enzymes take a different approach, using water to cleave peptide bonds. Pepsin, important for digestion, and HIV protease belong to this family. Metalloproteases, these metal-dependent cutters, also known as MMPs, are crucial for wound healing, fighting infections, and tissue remodeling. The benefits of breakdown. Protein catabolism serves several essential purposes. Recycling. By breaking down old proteins into amino acids, cells have a steady supply of building blocks for creating new proteins when needed. Quality control. Catabolism removes misfolded proteins that could interfere with cellular machinery. Cellular adaptability. This process allows cells to adjust protein levels quickly in response to environmental changes or cellular stress. Overall, protein catabolism is a highly controlled process that keeps the cellular protein pool healthy and functional, contributing to the well-being of the entire organism. Don't keep all this valuable information to yourself, share it with your friends who might find it interesting and beneficial. And if you have any questions or comments, feel free to leave them down below. We love hearing from our viewers and we will do our best to answer all your questions. And finally, don't forget to ask for our ASCP short notes to supplement your studying. These notes are a great resource to help you review and retain the information we cover in our videos.